Hi, my name is Yijia Li, and today I will present the CosTaL. It's our recent work, which is accurate and scalable graph-based cluster algorithms used for high-dimensional single-cell data analysis. So when we talk about analyzing the single-cell data, the very first question is to deal with the heterogenesis of the single-cell data, which means that there are dozens of types of cells. And in order to do a higher levels of analysis, we have to identify them first. And typically, there are three major types, which are manual gating, the supervised clustering, and the unsupervised clustering. And specifically, the unsupervised clustering is one of the most welcome methods due to the fast computing speed as well as its flexibilities to get easily integrated into more complex analysis pipelines. And additionally, the unsupervised clustering is also capable of detecting novel subpopulations. So all these good features really just makes the unsupervised clustering a very welcome methods. And based on the summary about how often they are used, we can see that among all of those very popular unsupervised clustering methods, the phenotype graph is one of the most prevail one. And if we take analysis about how the phenotype is able to achieve that, you can figure out that it's you taking the strategy about transforming the original high-dimensional space into a KNN graph. And then on top of the construction of the KNN graph, the phenotype will prune the graph, and lastly use the pruned graph for the cluster detection. And as a matter of fact, not only phenographs, there are also many other widely used tools, including Sorout, SCAMP, and also PRC. They're all formulating the clustering idea based on this strategy. But still, there are improvements to be made. And first one is the accuracy. Because even though the graph-based clusterings are performing very well, it's not always guaranteed to be the best performing one. And additionally, in some cases, the running time is very long, and especially for those large-scale datasets, which means that if there are higher dimensions and more cells inside, it gets more challenging for all these algorithms to be well compatible with those datasets. So based on the these shortcomings that are currently can be seen in the field, we designed the algorithms named CosTaL, which the full name is cosine-based Tanimoto similarity pruned graph, used for community detection by the latent algorithms. And similar to the predecessors, the CosTaL also takes 3 steps to just achieve the graph-based clustering. But specifically for the KNN mapping stage, we use the L2KNG, which is an ultra-fast KNN mapping algorithms that is developed by our co-authors, David and Natsu. And for the second step, being different from the others, we use the Tanimoto coefficient as the pruning methods. But lastly, we back again using the mostly welcomed latent algorithms for the community detection on top of the pruned graph. So I won't spend too much time explaining how the latent algorithm works, as of the comparables algorithms, they are using this. But I will just spend more time just trying to explain how the L2KNG as well as the Tanimoto coefficient really just empowers CosTaL to be both accurate and fast computing. So for the first step, which is to map the KNN graph, the fundamental question that are coming to our mind is like, what kind of matrices should be used to map the KNN? So here is a demonstration just representing if we are trying to find the neighbors of a vector A, which are representing one cell in its dimensional features. and there are B1, B2, and B3 as candidates. So if we are going to use the Euclidean distance, which are colored in blue, we will see that actually the B1 can be considered as the nearest neighbors, but B2 and B3, even though B2 maybe you can consider it's like more similar to A, the Euclideans just treat B2 and B3 all equal to like a same level of neighbors compared with A. But if we take use of the cosine similarity, we can figure out that actually B1 is still one of the closest neighbors, but B2 also is the other closest neighbors, as the angular distance from A to B1 and B2 are the same. But for the B3, as you can see here, it's not the top candidate of the nearest neighbors. So back again, both the Euclidean distance and cosine similarities are used for mapping the KNN, and as a matter of fact, these two are the most widely used. But we reason that the cosine similarities will be a better choice based on the fact that most of the single cell data, they're sparse, and also they are proportional data. Which means that the pattern of the features rather than the amplitude, they are the real contributors to how similar they are. And if we take the angular distance, it might just be a better fit. But still, the cosine similarities has its shortcomings. So the things we are trying to do is trying to compensate for this amplitude ignorance in the second step for the tiny multiple coefficient pruning. So back again for the reason why L2KNG is such an efficient KN mapping algorithms, I will just quickly walk you through how it works. So it took 2 steps for the L2KNG to return the KNN. The first step is known as the proxy step, which is to generate the candidates efficiently. And the second step is to verify the candidates to make sure that they are the exact neighbors. And for the first approximate stage, it's really empowering the L2KNG to be ultra fast. It fundamentally took use of 2 basic principles. The first one is like the high weight features count heavily towards the similarity of the 2 vectors, which means that if 2 vectors share a lot of those high weight features, they have higher possibilities to be the neighbors with each other. And the second strategy that it took use of is the fact that an object's neighbor's neighbor is also likely to be their neighbor. And because basically all these neighbors, they should be very same. And by just taking use of these 2 basic principles, the L2KNG just achieved the fast computing speed. And in order to see how it performs, actually we compare the L2KNG approximate with the other 2 approximate KN mapping algorithms, the NN descent, the greedy filtering on 4 datasets using parameter k from 10 to 100. And as a result, you can see that L2KNG approximate is performing better than the other 2 approximate methods. And if we add into those the L2KNG, which is the exact mapping algorithms, you can see that it's performing very competitive with the others. And altogether, this just proves that the L2KNG is a very powerful KNN mapping algorithm. So once we have construct the KNN graph successfully with L2KNG, the next step is to print the graph. So back again to the previous question that we have, which is the cosine similarities, they cannot map the amplitude difference. And based on that, we use the time coefficient to replace the edge weights of the KNN graph, which used to be the cosine similarities. And by taking use of the time coefficient defined as this, we can just bring back the amplitude information. But in comparisons, the other methods, including the phenograph, the Thoroux, and the PRC, they are using the J-class similarities to prune the graph. And basically, these two are different strategies, which the Tanimoto coefficient is trying to restore more of the original similarities, while the J-class similarity is basically finding the similarities by its neighbor information. So other than these fundamental differences, we find that using Tanimoto coefficient really benefits the computing speed. And in order to evaluate how the CosTaL performs, we use the other existing graph-based algorithms against CosTaL for the comparisons on two data sets. One is for the mass spectrometry, and the other is from the single-cell RNA-seq. Both are considered to be large scale, which have a lot of cells inside. And for the results, we focus on the computing speed, the number of clusters generated, as well as the precisions compared with the manually labeled, manually gated labels. And in order to be comprehensive, we use 6 different matrices for evaluating the precisions. And here are the results that are generated. The topper ones are for the mass spectrometry dataset, and the lower ones is for the single-cell RNA-seq datasets. So for the number of clusters identified by all those algorithms, we can see that actually the CosTaL is generating very similar results as the others. But for the computing speed, CosTaL is outperforming the others. And as a matter of fact, if we take a look at the single-cell RNA-seq datasets, the CosTaL is able to achieve the fast computing speed using the 2000 highly variable genes as the features, while the similar speed can only be achieved by scampi and PRC using the PCA transform data, which is only 50 dimensions as the features. And for the precisions, the CosTaL is performing very similar with the others on the mass spectrometry datasets, but the CosTaL is performing better than all the other algorithms on the single-cell RNA-seq datasets. And then it brings us to the conclusion, which is, firstly, CosTaL is ultra-fast and scalable. And also, it can generate high precision results in compare with the existing tools. It's very competitive, and sometimes even it's performing better. And lastly, for the sequencing datasets, actually you do not need to use the PCA to achieve fast computing speed as well as the high precision scores. And as a supplementary, I provide the process about using CosTaL against the other cluster methods for actually analyzing the single-cell RNA-seq datasets. And as you can see that CosTaL is really shortening the analysis process, and which generally means that it can iterate faster than the others, especially will be beneficial to large-scale datasets. And that's all I would like to share today. And thank you so much for listening me through. If you have any question, please just reach me out through my email, and I'm happy to communicate with you all. And thank you so much.